Welcome back to my channel Knowledge Chronicle.
Have you ever wondered how the language of life stored in our DNA turns into action? How our cells understand the genetic blueprint to make proteins that build and run our bodies? The secret lies in a process called transcription, the very first step in decoding life. Welcome to Knowledge Chronicle, and today we'll uncover the fascinating journey of genetic information from DNA to RNA. Buckle up, because by the end of this video, you'll see just how incredible and precise nature really is. Transcription is the process of copying genetic information from one strand of DNA into RNA. Think of DNA as a thick book of instructions, and transcription is like copying a specific recipe onto a sticky note so you can use it in the kitchen without carrying the whole book around. Now here's the twist. Only one strand of DNA is copied, not both. Why? Well, if both strands were copied, the RNA sequences would be different due to complementary pairing, leading to confusion in protein production. Also, the two RNA strands could pair with each other to form double-stranded RNA, making it impossible to translate into proteins. So, to keep it simple and accurate, only one strand is transcribed. A transcription unit is like a mini blueprint on the DNA. It tells the cell where to start copying, what to copy, and where to stop. It has three major parts. 1. The promoter, the starting point or the green signal for transcription. 2. The structural gene, the part that actually gets copied into RNA. And 3. The terminator, the stop sign that tells the cell to end transcription. Here's where it gets interesting. DNA strands have direction, like one-way roads. They run in opposite directions called polarity. One goes 3' to 5' and the other 5' to 3'. RNA polymerase, the enzyme that copies the gene, can only read in one direction, so the 3' to 5' strand is used as the template. But the strand that doesn't get copied, surprisingly, is called the coding strand. Why? Because it has the same sequence as the RNA being made, except it has thymine instead of uracil. Let's take an example. Template strand. 3' HECAT GCAT 5'. Coding strand 5'. Tag 3'. RNA transcribed 5'. UAC guac 3'. Notice how uracil replaces thymine in RNA. We often say it's in your genes. But what exactly is a gene? A gene is the functional unit of inheritance. It carries the instructions to build a polypeptide or a type of RNA. In bacteria, a gene often codes for more than one protein. These are called polycistronic genes. In humans and other eukaryotes, each gene usually codes for one protein called monocistronic genes. But wait, genes in eukaryotes are split. The coding regions called exons are interrupted by non-coding regions called introns. Imagine trying to read a sentence with random words inserted in between. That's what raw RNA looks like. So, the cell splices out the introns to stitch together the real message. Also, don't forget the promoter and regulatory sequences. They don't code for proteins, but they control the activity of genes, like the volume knob on a speaker. Now let's talk about the three key types of RNA. The superheroes of the protein-making world. Messenger RNA, or mRNA, is the blueprint that carries genetic information from DNA to ribosomes. Transfer RNA, or tRNA, is the supplier that brings in the right amino acids during protein building. And finally, ribosomal RNA, or rRNA, is the architect and builder that forms the structure of ribosomes and helps assemble proteins. Each type plays a unique role, and together, they make the magic of translation happen. In bacteria, life is simple but powerful. There's just one enzyme, RNA polymerase, that does it all. It goes through 3 stages. First, initiation, RNA polymerase binds to the promoter region. But it needs help. An initiation factor called sigma helps it recognize where to start. Next, elongation. RNA polymerase reads the template and adds RNA nucleotides one by one, following the base pair rule. Finally, termination. Once the RNA polymerase hits the terminator sequence, it detaches with the help of a termination factor, often called RO or Rho. A fun fact? In bacteria, transcription and translation happen simultaneously. As the RNA is being made, ribosomes jump on and start translating it. That's efficiency at its best. Eukaryotic transcription is like a grand orchestra with more instruments and more control. 3 types of RNA polymerases exist. First, RNA polymerase W makes rRNAs. Second, RNA polymerase II makes mRNA, actually a precursor called hnRNA. And third, RNA polymerase III makes tRNA, 5S RNA, and snRNAs. Once hnRNA is transcribed, it undergoes three crucial processes. First, there's capping. A special hat called methylguanosine triphosphate is added to the 5' end for stability. Next, we have tailing. A tail of 200 to 300 adenines is added to the 3' end, and this is called the poly-A tail. Finally, their splicing introns are removed, and exons are stitched together to make the final mRNA. This polished mRNA is now ready to leave the nucleus and make its way to the ribosomes for translation. You might be wondering, why all this complexity? The answer lies in evolution. The presence of introns and RNA processing may be remnants of an ancient RNA world, a time when RNA ruled before DNA and proteins took over. This complexity also allows for alternative splicing, where one gene can make multiple proteins, increasing diversity without increasing the number of genes. From a single strand of DNA, nature writes messages in RNA, edits them with precision, and uses them to build life's machinery, proteins. The elegance of transcription is a powerful reminder of the intelligence wired into every cell of your body. If this deep dive into transcription sparked your curiosity, let us know in the comments. What topic should we decode next? And remember, your one like, share, and subscribe will help us create more valuable content.